live here in a few minutes. We'll just give it a couple minutes to buffer, but yeah, so recognizing we do have some student or some audience members in the class. Perfect, there we go. So, hello everyone, my name is Amanda Piper. I am an education coordinator at the Canadian Light Source. Welcome to Students on the Beamline seminar, brought to you from students from Vanier College in Montreal, Quebec. On behalf of the students, as well as CLS, I would like to acknowledge that the Canadian Light Source, where this experiment took place, is located in Saskatoon, Saskatchewan, in the traditional homeland of the Métis, as well as Treaty 6, and the homelands of the Nehawk, Anishinaabe, Lakota, Dakota, and Nakota nations. And like I said, on behalf of the team, as well as CLS, we would like to acknowledge that we respect Indigenous ways of knowing and oral traditions. And throughout all our partnerships and relationships across country and across the globe, we would like to acknowledge that we're moving forward in a spirit of partnership, reconciliation, and collaboration. And we are thankful for the land that we are able to work on. So just a couple housekeeping things before we get to the main show. We are live streaming through YouTube, so feel free to keep your videos off. I believe the students are going to be sharing a recording as well, so that might help keep your videos off. To save on bandwidth. For those of you that are joining in Zoom, you do have the option to have a different layout. So if you notice in the right-hand corner, there should be a view icon. Feel free to change, play around with that as the students will be speaking, presenting from their classroom. And I'm not sure if you caught, but they all also have guests in their classroom as well. So we'll be speaking to those guests as well as to us in the Zoom call. We do have a question period at the end. So if you do have questions, feel free to post them in the chat or raise your hand or unmute yourself if you're in the Zoom call and state you have questions and wait to be called upon so that the students can manage questions both from their audience as well as from those in the Zoom call. And yeah, if you, Gabriel, want to get started with sharing your screen, I'll just finish introducing you. So like I said, these are students from Vanier College in Montreal, Quebec. They used the Ideas Beamline virtually a few weeks ago to investigate or examine the roots, stem, and leaves of 3 different plant species that they collected from their gardens on their campus grounds. And they were interested in looking for patterns in phytoremediation, as well as hyperaccumulation of cadmium, zinc, and other few elements of interest. So it looks like we're almost there. I can see your slides, Gabriel, but you just need to switch it into presentation mode. So if you go to the top at the display settings, yep. So just at the...
Yeah, because I clicked present.
Yep, if you go there and you should be able to switch. Yep, swap presenter view or... Perfect, does that work for you? Yeah.
Yeah.
All right, the floor is yours.
However, you have, oh, next slide. Amanda, can you see just one slide or can you see the next slide as well?
No, we can see one slide. We see presentation.
Well, I guess it's up to you. I guess if you go to display settings, you can still change it.
Because the display isn't quite all right for the classroom.
Because it's just the next slide. It's not.
But will the animations work?
Oh, I guess it will show the notes as well. If you go to display settings. Oh. So you need to do this case.
Going back to display settings like Lysimachia is talking about, if you just duplicate it, I don't know if you'll be able to see the notes, but whatever you see is what will be shown for both screens.
And then display settings. Yeah. Oh. I don't know how to fix that.
I just want to know slide and see if the animation works.
Oh.
It works there.
Yeah, maybe that's good. No.
Try going to one slide and see if the animation works.
And just duplicate.
No, it's good.
That's it.
That's good. Look at that.
Can you hear me properly?
Yep.
Start. Good. Yes. All right. So, hi, my name is Gabriel, the elected team leader of this group project of students on the Beamline, where we got the chance to use the Beamline from the Canadian light source Synchrotron, a particle accelerator, to conduct the study of our choice, which was cadmium and zinc hyperaccumulation in Lysimachia, Nummularia, Achillea, Millefolium, and Symphytum Officinale. I'm a fourth semester student in health science, and it's my second time around working on a project with the CLS.
Hello, everybody. My name is Maria. I'm a first year in health science. And the reason that I joined this project is to have an insight in the research project and more insight about the science field.
Hello, my name is Jamil. I graduated last semester from Vanier in health science. I wanted to continue in this research because it gave me an insight of what to expect in university and how to continue through my life in university in the field of research and science.
Hello, everyone. My name is Diana Lacar. I am a second year student at Bani in music and science. I joined this research team because I wanted to gain some experience in a research project led by students.
So hello, my name is Jade Desjardins. I am a first year health science student, and I joined this project because I'm interested in working in the field of research in the future.
Hi, my name is Nandita. I'm completing my last semester here at Vanier in health science. And I joined this project since I wanted to explore research as a potential field for my future.
Hi everyone, my name is Lalay, and I want to be a dietitian. That's why when I saw this opportunity, I grabbed this right away because I want to gain more experience in the research field. But before we start our presentation, we acknowledge that the place in which we work and study is situated within the traditional unceded lands of the Ganyan Gahaga, Mohawk peoples, part of the Haudenosaunee Confederacy. There is also a strong historic presence of Anishinaabe peoples in what is now known as the Greater Montreal area. Yotoge, or Montreal, has also long been and continues to be a gathering place for many First Peoples from all directions. We honor and thank the traditional custodians of this land and strive to work for the success of future generations. We also want to honor the land of the Canadian light source, which is shared by Amanda.
Maybe we can minimize this so that it's not.
So yes, I'll start. So Vanier College is located in Montreal, Quebec, following the Quebec educational system. For those in Saskatchewan, how it really works, well, there's 5 years in high school, which is the equivalent of grade 7 to grade 11. And after that, students go often in college, which brings up the age between 17 and 18. They go for 2 years or 3 years. 2 years for the pre-university, so they'll complete 2 years and then go to university, or 3 years where they specialize themselves in a career and then pursue a career after college. There's roughly 6,000 students here. And so CLS Beamline collaboration with students refers to them, CLS, the Canadian light source, and us, this is our project.
So our campus, Vanier infrastructure consists on 9 buildings, which we mostly identify them with the letters. So everybody knows where we can go. There are 3 parking lots, and our location is pretty interested because we are in the middle of two graveyards, which is only a fence that they divide us. Around all the campus, there are some planters, which is the gardener who is in charge of it. However, in summer, there are some students who work in it, and the gardens have a specific purpose of them. For example, they are medicinal gardens, they are biodiversity, they are animal habitats, they are open gardens. But in this project, we're going to refer to the gym location, which is the G that you can see, and the planter that is behind the K&D building.
Part of our story was finding the right research question. So we started off in December, and then after many months of reformulating the question, brainstorming, lots of literature review, in December, we settled on a phytoremediation and hyperaccumulation study. Take a look at this tree. The white lines on the bottom represent the contaminants and heavy metals absorbed by the tree and stored in its tissues. Our goal here is to radiate these tissues with X-rays and then study the metals present within them as a result of the contamination. Part of the reason we settled on this angle project is due to its environmental focus. This project deals with contaminants present in nature and its effect on trees and wildlife. Another reason is because it tackles local issues. The plants we chose are from our very own campus grounds, part of the Vanier Gardens. The Vanier Gardens, which include many plants which are readily available for student consumption. So it's extremely relevant to monitor and study the elements present in these plants. Those are personal reasons why our team chose this project, but there are also more broad reasons for studying phytoremediation.
So as Gabriel mentioned, we focus on an environment and in plants. And we got into thinking that first that what methods there are to reduce pollution. Like what methods can be used to reduce the pollution that is around us. As he mentioned, like the plants that they are planted here, they are consumed by the students, which can be hazardous if they have any contamination to our health. So for this reason, we obtain scientific data to support permaculture. Permaculture idea came up from the gardener, Mark. He was the one who started saying that permaculture is an agricultural method that they use by using other organisms to support each other to maintain a self-sufficient ecosystem and as well as sustainable. By this idea, we got into bioaccumulators because he mentioned that there are some plants in Vanier gardens, they can be defined as bioaccumulators. A bioaccumulator is one of the plants that be able to uptake heavy metals from the soils to decontaminate by storing these elements in their leaf tissue. For example, at first you have this plant. You have the plant here. And this plant has been around the industry, and this industry may spill some chemicals onto the soil. As you can see, the roots will be the first plant organ that will absorb the heavy metals and then distribute them and store these heavy metals through the leaves and stem. By having this idea and start digging out the literature review on all these idea of methods to reduce pollution, we got into phytoremediation. Phytoremediation is a technique that used to decontaminate specific soils by using plants. By having this phytoremediation, we might not only benefit us the environment, it also benefits the economy because it's low cost by just using plants, as well as having our health benefits that are not giving us conditions of having this exposure towards these heavy metals. Then we got into the research question. Can Lysimachia, comfrey, and yarrow hyperaccumulate heavy metals such as cadmium, zinc from the soil in a location? So at first we identified the plant species that were in the garden. We found there are many species in our gardens such as dandelions, comfrey, Lysimachia, and yarrow. The ones that we chose was based on the literature review that we found out that these plants they have the heavy metal that they mostly absorb is the zinc and cadmium. Why we focus on these heavy metals is the first question. Because these heavy metals in a large amount, they might get us exposure, giving us some new conditions to our bodies. For example, they might get into gastrointestinal diseases. It has been proven that in homelands of Canada, they have these diseases due to the condition of exposure of cadmium and zinc. So we got into the hypothesis that this Lysimachia, comfrey and yarrow, will absorb cadmium and zinc from the soil in which these plants will be able to store and uptake the heavy metals. At first, the plant species will be considered the scientific name as Achillea millefolium. We will commonly use it as yarrow in our research project. Lysimachia Nummularia, it will refer as Lysimachia, and Symphytum Officinale, it will be referred as comfrey. All these plants are perennials, which means that they survive through the winter. They adapt easily to any temperature and over the regions. Comfrey and Lysimachia, they are not North American native plants, so they come from Europe. However, small pieces of Lysimachia, they just can easily attach to the soil, and they start reproducing themselves. All these plants, they also share the condition that they might be useful to medicinal purposes. They are used for doing teas or ointments to reduce some symptoms of some health problems and conditions. For example, comfrey, they might be used for any fracture or inflammation that may cause through the bones diseases. Lysimachia is often used like for the skin rashes, and yarrow might often use it for stomach disorders.
Now, let us look at plant physiology. A physiological basis will help us to understand why certain elements are found in our plant samples. Okay. So, metallic elements such as iron, zinc, and copper are required for essential metabolic processes in plants, such as photosynthesis, respiration, enzyme synthesis. For example, as you can see here, the electron transport chain. These elements are vital for it, and this chain produces energy. And now this energy is again used in these metabolic processes. Furthermore, for example, iron is used in the synthesis of chloroplast, which is the site of photosynthesis, and zinc in the making of chlorophyll, which is the green pigment, also essential for photosynthesis. All in all, to show that these are vital for plant survival. But, however, although vital, zinc in excess can be toxic. It can inhibit these essential processes in plants that are not adapted to these excessive zinc conditions. And the plants that are hyperaccumulators. And not only to plants, this can also be toxic to animals and humans through the process of biomagnification. And in contrast to our essential heavy metals, we also have cadmium, which is not an essential micronutrient, which means it has no nutritional value for the plant. Therefore, its presence in our data would suggest that hyperaccumulation of this toxic metal takes place.
So as I mentioned before, there are two planters that we were focused on, which is the gym building and behind the deep building. All these planters have some variables that they share. For example, they are often used to circulate students over there, and they are consumed by the students as well, these plants. This planter has other variables that they don't share. For example, the exposure of the sun during the whole growing system, because as you can see behind the deep building, there is like this wall that you can see on the picture, which means that that's the wall from the building. Basically, the building covers the whole planter, which gives more shadow than exposure of sun. In the case of the planter of gym building, they have more exposure for students and also for the sun. Both buildings both planters, they have a parking lot beside them, which it might be contaminate the plants by going into the behavior of these plants as they might respond to the uptake of heavy metals. You might wonder why there's no snow on the pictures, and it says at the bottom, pictures taken on December. Well, that day that we took our samples from these planters, it was a warm day and it was rainy. That's why you are not seeing that much snow. But as you can see on the corner, there's a small white piece of snow because you can see the snow is still there because it was winter. But since it was rainy, the soil, it was wet and the plants were still green because it was just the beginning. And as I mentioned, the plants are perennials. So they survive to the winter. From these planters, we took out all the plants that they were there. However, behind the building, we just found 2 species of them. How did the plants look? So for yarrow, you can see that this one was taken from the gym garden. You might be wondering why there's like a lot of leaves over and everything. Well, that, as I mentioned, it has more exposure to the sun. That's why you can see the leaves are less green than the other picture that is behind the building. We might deduce that these are not often exposure to any other conditions like the gym garden. They still green, as I mentioned, because they are perennials. And there's a difference between the colors from the Lysimachia of both buildings. As you can see, Lysimachia here is green. On the other side, you might see there's like a small purple parts of the plants because there are many different species of Lysimachia. But in this case, both of them are the same species, but not all of them have the same colors. Have been found the same species. They might be in yellow leaves that we saw in our garden. There were some yellow leaves, and there were also other ones that were purple and brown colors. The soil was still on the surface. It was just wet, but into going deep, the soil was frozen. So this sample, we took the plant species from every planter. Behind the BD building garden, we divided into 2 sides. So we can just contrast both of them if they hyperaccumulate the same way, either if they have different sides of the buildings. The yarrow and Lysimachia, they were in both locations, but the comfrey there wasn't on the gym garden. For the surface soil, we will refer the soil as surface because since the soil was frozen, we couldn't go as deep as we wanted. It was just around 10 to 15 centimeters as depth, which is mostly referred as surface soil rather than soil samples. And the collection day was in December 16 in 2021. So our sampling method and procedure was based on the literature review that we did in experiments that were based on phytoremediation. The plan consisted on collecting the plants and soil samples from each planter according to the species. How we collect them? First, we have to identify the plants from each one according to their physical characteristics based on our literature review. Then we store them in Ziploc bags. The Ziploc bags were labeled in a code system that we came up. It was with the plant species that were sampled and what the location. Then we store the plant samples in the freezer at minus 20 degrees Celsius until we were going to plan to take them out so we can do the lab work. Then starting in January, we start doing the lab work and we wash the plants with tap water and ionized water. We start removing the dead material because since they were too long, like almost a month on the freezer, there were some dead material on it. So we had to take it out. Then we start cutting the plants into small pieces so we will be able to open dry them later. We place them in samples in Petri dishes, and we dry them in an convection oven at 82 Celsius degree, and we left them like for 2 days until they were completely dry. And how we determined that they were completely dry, because our literature review was based on that the plants will be stiff, they will be rigid, and the material will look darker. Then we start crushing the samples with a mortar and pestle until they were looking into a powder characteristic. After we place this powder that we put into test tubes, again, we prior labeled the test tube with the same codes as the Ziploc so we were able to identify them later. We placed the samples and we did some replicas on it, and we put them into sample holders. Here in the sample holders, we realized that we were missing a sample, but it was the tap water. It might influence our plants and the whole procedure because we wash the plants through them, so it might influence on it. That's our first thought. However, our results said the difference. Then these samples travel into the other province to the CLS, and they were into the Beamline, into the Synchrotron, where they were going to get our data results.
Okay, so what is Synchrotron? Well, first, it has the purpose of producing a very bright light, and this bright light can go at different wavelengths, and we'll refer to this bright light as synchrotron light. Scientists used the synchrotron to examine structural and chemical properties that are invisible to the naked eye or with other tools. So this really lets us go more in depth inside. So how it all works? Well, it all starts with the first point, which we can see here on this first point, which is the electron gun, which heats up and releases the electrons towards the linear accelerator in the second point, which, as the name implies, it accelerates the electrons towards the booster ring, which is the third point. And the characteristic of the booster ring is that it's circular, has a high vacuum, meaning has very little matter, and it's a steel tube. So the electrons travel in a circular motion in the booster ring. Once they've reached at a certain energy and the speed almost equivalent to the speed of light, they are transferred to the storage room, which is the fourth. At the storage ring, they have two paths that they can take. First one is that they're going to continue in a circular motion that are caused by very strong magnets that forces the electrons to continue turning. And the second path is a straight path, which is where the electron will deviate and will go in beamlines, which you can see here as the red lines. These are different beamlines that are present. So the fifth point is the synchrotron light. So this deviation of the electron will be used as synchrotron light. And then, as you can see, point 6, it's the beamline. So the CLS has 22 beamlines. We use the IDEAS beamline, which is specialized in students' projects or other educational purposes. Then the seventh point is the optics hunch. And then the synchrotron will go travel into the experimental room, which is where Dr. David from the CLS had placed our samples. And then all the data was collected in the control room. X-ray, well, XRF. So the synchrotron is a cool tool, but there are different techniques that you can use. One of the techniques that we used is XRF, which it allows us to determine what is present and absent in the elements, in the samples that we had presented. It all starts with the X-ray that is directed at the sample. And the idea is that the X-ray is energetic enough to excite the electrons inside the atom. My teammate Jade was kind enough to make a really cool drawing here. So we have the nucleus in the middle and then different shells. And the idea is that the little dots around is electrons that are just hanging around on different shells. So X-ray is going to come, it's going to be energetic, it excites the electrons. And now the electrons are either promoted to a higher shell or they're emitted from the electrons, from the atom. When it is emitted, another electron from a higher level, higher shell, has to drop down to fill in that hole, that space. When it does that, it releases energy. That released energy is going to be detected by the beamline, and that's where our data is based off.
So when it comes to understanding what exactly we're seeing in XRF graphs, we actually have to understand how to read it first. First, I have to know that the XRF technique is qualitative. It is not quantitative. You can't measure mass, you can't measure percentage, and you can't measure quantity of an element within a sample. All you know is which element is present. Now, when you look at the graph here, you can see that there's multiple peaks. Each peak represents an element, and each element is comprised of 2 peaks. So this peak here is iron on the top, and this one as well. And they have a secondary peak right here, still iron. It's just that this one is the K alpha, and this one is the K beta. The K alpha represents the peak where the electron jump occurs most commonly due to the lower energy required to do so. The K beta is the electron jump that still occurs, but a little less frequently due to the fact that it requires more energy. You also have to understand that when we're looking for the energy at which an element initially appears, you have to look at the K alpha peak, because that is where the range begins. Also, when we want to determine an order of mostly absorbed elements within a sample, we have to look at the K alpha peaks due to the fact that they are typically the highest ones, as I've said before. If you want to determine the height, you have to look at the photon count right here, which is on the y-axis. So also, when you want to input emission lines to identify which elements are present, the solid lines represent the K-edge emission lines, as was mentioned earlier by Diana, and the dotted line represents the L-edge emission lines. Now, as you can see, I circled here actually the energy range, the electron volts range that is used during the XRF scan. It requires a broad range of energy because each element is excited at a different energy. Each element fluoresces at a different energy. And we need to have as broad a range as possible in order to detect as many elements as possible for the ideas beamline specifically. Now, another beamline may also use XRF, but the equipment may be different. It may be more precise. Maybe it can use more energy, less energy. So the results may not always be the same. So onto the results. When we're looking here, we see that Lysimachia, comfrey, and Yarrow absorbed iron, calcium, copper, and potassium. Now, I want to say that there is copper present in all the plant samples, but the peak is very small, but it's still clearly identifiable, as you can see here. Something that we should have expected, but we'd never initially mentioned in our permit template, was that the plants all accumulated relatively high amounts of iron, calcium, and potassium, which is normal since they are elements necessary for plant function. Now, for the next slide here, you can see that the 2 graphs represented are the left and right sides of the debuilding for the leaves of each plant. You can see that comfrey absorbs more zinc, a little bit more zinc than the other plants here. The blue line is the comfrey for both left and right. And that yarrow right here absorbs overall, when you look at the 2 graphs, more iron than the 2 other plants. Now, for the other slides, which are the stems on the right and left sides, you can see that comfrey absorbs the least of all plants in iron. And this is contrary actually to our literature, because what we saw was that comfrey was supposed to absorb the least zinc and cadmium, and its stems and leaves specifically were supposed to absorb higher amounts of iron. Now, taking a closer look at the graphs that we just saw before, we can see that generally there's a similar absorption within the plant parts, but once again, comfrey is one that stands out. As you can see in the stems, it absorbs half the amount of iron that is present in the leaves and double the amount of copper in the leaves. with the zinc having very little to no change, but still remaining the one that absorbs the most out of all plants. When we look at the soils, there is the G soil and the D soil. We chose the left soil as we are focusing mainly on this side. There's very little to no copper in these soils. Now, this may be because we have only surface soil, or this may be just because there's not much in the soil at all. But what we notice is that the photon count hovers barely above 0, so much so that when we initially looked at the samples, we didn't identify copper at all. However, when you zoom very closely, it just hovers a bit over 0 for the photon count. Another thing to notice is that our initial question was about cadmium and zinc. Well, where's the cadmium? There is none. So our soil was never contaminated with cadmium, which means that we can't actually have an answer for a question. So our results, specifically with our research question that we had at the beginning, are inconclusive. Now, while we initially decided that we wanted to compare the absorption levels within the plant samples and the soil samples, it was determined that we can't actually do that because we don't have consistent masses within our samples. It just wasn't possible for us to have a good amount of volume for the scan while still keeping the same mass due to the different densities of each sample. I do want to point out here that when looking at the graphs, as the colors correspond to the order that this elements are selected in, there is a slight discrepancy here. The argon is not actually zinc, it's argon, it's just not red. And then from then on, every other color has also changed.
There are possible explanations for the elements present in the sample. Cadmium, as Nandita mentioned earlier, isn't an essential nutrient for the plants. So if there was no excess contamination of cadmium, that would likely explain why it was not in our samples. Zinc is often used in fertilizers. Considering these plants were planted by a gardener, that's likely or possibly where the zinc originated from. Potassium was also an element present in many samples, and it's also present in fertilizers. And iron, as many of us know, is very, very rich in soil, which explains why it was often the highest concentration, especially in the soil samples.
So ZANES, just like XRF, ZANES is another technique that you can use. Now the difference between ZANES and XRF is that ZANES was more specific down to one element. So if you remember correctly, XRF is generally what element is present inside our sample. XRF is what what molecules is specific in the samples. So this took a little bit longer because now we had to tune, in a sense, the synchrotron to a specific element for it to detect the element that we were looking for. This means that the range will be more specific, so it will be a little smaller. Another difference is that the XRF scan took roughly between 3 to 5 minutes, depending on the density of the samples. Obviously, soil was more dense than, for example, the leaf samples that were a little lighter. While the XRF, it took roughly 15 to 20 minutes per sample. Thus, we didn't have much time to go over all the samples. We prioritized a few samples that seemed to be important.
So Diana just explained what ZANES is, but what would you do with a graph like the one on the right, for example? What would you do with the ZANES graphs? So it brings up, we're not looking for a specific number. We're just looking at the graph by itself, by a whole. So it's rather a qualitative method rather than quantitative. One of the main functions of the ZANES graph is that it shows the different forms of a specific metal. Now, as Jade explained, our initial question contained cadmium and zinc. But since we couldn't find any cadmium, we concentrated all the ZANES graphs on only one metal, which was zinc. Now let's look at the graph on the right. As you can see, the probability of excitation increases sharply when the photon energy matches the binding energy of a core electron. That means is that it's always normal until we reach when the photon energy reaches the binding energy, we have this huge jump over here. Well, one of the main or one of the very important reasons that ZANES work actually is that the binding energy of core electrons in various metals is well separated. As ZANES talked about, as Jake talked about in the XRF scan, we have various metals in different energy levels. As you can see, we have iron, we have zinc, we have bromine. They're all in different ranges, and that's why it's ideal for us to use ZANES graphs to compare the same metals. Now, the ZANES graph of same metals are compared after the main edge. The main edge is after the first jump. So after the first jump, we can compare the same metals, the ZANES graph of the same metals to figure out the different forms of that metal. Okay. So there's different forms of zinc. So some examples are zinc nitrate, zinc oxide, and even zinc sulfate. So looking at the ZANES graph, after the main edge, after the straight line, we can start comparing the different trend lines of the different forms of zinc. And looking around inside the circles, we can see that every trend line has a very different form, which means, as you can see, that those are all different forms of zinc. In an ideal situation, what we would have done in an ideal situation is that we would use the ZANES graph of the sample we have of zinc, of the plant we're looking at, and then we'll compare it to the zinc standard zinc graphs of the different forms of zinc, such as those on the right, and then we would compare the trend lines of those two ZANES graphs to figure out what form of zinc we have in those data. However, in our data shows a mixture of zinc-containing molecules, making it hard to exactly compare, but rather to estimate what forms of zinc we have in our samples.
So you've seen the XRF graphs, and now it's time to present the ZANES graphs. Again, as Jamil said, all about the zinc. We'll be focusing on four main comparisons. First, between the three different plant organs. Second, between the planters. Third, between two different plant species and the soil in the D building. And we'll do the same for the soil, for the area in the gym. So starting off with our very first comparison. This is to answer the question, do plant organs contain the same zinc molecules within them? Looking at the graph, you can see all three of them practically superimpose each other. They're almost identical, almost the same line. What this tells us is that the mixture of zinc species present, zinc molecules present in our samples seem to contain the same types. Next, do plants from different planters have the same zinc molecules? Let's start off with the yellow. The red line represents the molecules in the D building, and the black line those from the gym. Again, same story. After the main edge, they're practically the same. Now for Lysimachia, once again, following the main edge, they're practically the same zinc molecules. So to answer the question, yes, there do seem to be similar zinc molecules present in both areas for the plants, regardless of where they are. But what are the possible zinc molecules in the plants? Jamil said we could compare the molecules to standard values. So let's try that with the yarrow leaf. The yarrow leaf starts off with a rather smooth downfall after the main edge. And that's vaguely similar to the zinc bromide one, although it lacks the little bump at the end. So if we look at zinc ion, zinc 2+, that has a higher bump. So it looks much more similar to the zinc ion. But what's most interesting here is that zinc ions are apparently absorbed by plants. Plants use them for the metabolic processes that Nandita mentioned earlier. And perhaps even more conclusively, the Lysimachia roots have a shape that resembles even more the trend line of the zinc ion. It has that little chair shape in the middle. Continuing the comparisons between the planters, do the soils differ? So there we have the orange line representing the gym, the green line representing the D building. And immediately you can see that, yes, they do contain different zinc molecules. If we see this first circle, the green line clearly goes underneath and has a different shape. And over here, the orange line clearly goes above.
So to continue to talk about what Gabriel was saying, this ZANES graph is just a combination of all the graphs he just talked about. So the dotted lines are the yarrow plants in a different location, the D building and the gym, while the solid lines are the soil in the D building and the gym. So if we're going to start comparing, let's look after the main edge. We can clearly see, as Gabriel just explained, that the soils are different. So the trend line of the soils are different, which means that there's different forms. There are different forms of zinc inside those soils. However, if we look at this at the trend line of the yarrow plant, we can clearly see that they're almost identical. They're the same, which indicates that they have the same form of zinc inside of them. Now, a question arises: if you have two locations, and in each location there's the two different soils, they have different forms of zinc. However, the plants in those both locations have the same form of zinc. So, could plants be absorbing zinc molecules selectively? Well, as Gabriel just explained and showed it to us, that the yarrow plants and the Lysimachia had a very close, the trend line of those ZANES graphs almost had a very close trend line to the ionic form of zinc, the Zn2+. So in this case, both yarrow plants in both locations contained the ionic form of zinc, the Zn2+, where the soil, however, the soil where they were at had different forms of zincs.
Now, as we did for the plant samples, let's have a look and try and identify the zinc molecules present in the soil. Let's start with the G building. Its trend line is very, very similar to the red one here on the right, representing the zinc nitrate molecule. So here there's an orange line representing the orange trend line. It's pointed to the zinc nitrate molecule. Looking at the D building, its shape is strikingly similar to the zinc sulfate trend line. So it appears that in the gym, there was a much higher concentration of zinc nitrate in the mixture of all the zinc molecules. And in the D building, there was a much higher concentration of zinc sulfate present in the mixture of zinc molecules.
Now, we already compared the different parts of plants. We compared the same plants at different locations. So now we're going to ask the question, could different species of plants, such as yarrow and Lysimachia, have the same zinc molecules? So if we start looking at it after the main edge, we can clearly see, again, that the soil had a different trend line than the other plants, which, as we talked about and discussed, that yes, the soil had different forms of zinc in it. However, the trend line of the plants of the yarrow root and Lysimachia has a less of a difference between each other, which indicates, or we can hypothesize again, that they have almost the same form of zinc inside of them. So this all comes back to the same idea of what Gabriel said, that they all come close. If we compare them to the standard forms of standard forms of zinc, the ZANES graphs of them, they're very close actually to the zinc, the ionic form of zinc, the Zn2+. And this all supports a study done in the University of Minnesota, where plants actually tend to take the ionic form of zinc, the Zn2+. So to answer the question here, yes, the different species of plants also contain the same form of zinc. This ZANES graph is exactly the same as the previous slide. However, the only thing that changes is that here we're looking at it in the gym location. Ideally, we would have compared the yarrow roots to the Lysimachia roots, but as Gabriel said at the beginning of the slides, that the yarrow plant, all the different parts of the yarrow plant absorb the same form of zinc, which means it doesn't matter what parts we take and compare. So this also supports the same idea where the soil is different, the soil trend line of the soil is different than the plants, while the plants are almost identical or very close to each other, and also compares and goes back to the same trend line of the ionic form of zinc. So at the end, all of what we did compares back to the same or supports the study done in the University of Minnesota, where the plants actually are selective in taking are being selective of the form of zinc they're absorbing, even though the soil had different forms of zinc in it.
Moving on. So improvements and future experiments. As students researchers, we may not have a lot of experience in this kind of experiment. So we think that it's very important to address the limitations that we encounter during our experiment. And by doing so, this can help us improve our future research. So the first limitation that could have impacted our findings is the limited amount of our samples. As you guys can recall, we only use 3 samples for each of the plants. So if our sample size is insufficient, our study will not really produce a meaningful result, and it also increases the margin error of our research. This is because statistical tests requires bigger sample size to produce accurate results. And why don't we have a lot of samples? And as you guys also may have known, like we spent a lot of our time during the spring break doing this, and with the help of Dr. David Muir, and he spent a lot of his time like putting the samples inside the sample folder, and it all actually took like a day. So if we're gonna do like, let's say 100 replicates, it might not be possible right now. So secondly, there were some sample issues. Some soil samples were mixed with little bits of rocks. So the masses of our samples were not evenly homogenized. Ideally, all of them should have the same size. And as you can see in the picture, the soil should also be filtered so that the samples are evenly mixed. Moreover, deeper soil was undersampled. We obtained our samples during the month of December, where the soil was frozen and hard to collect. And as a result, our soil sample were obtained 10 centimeters below the ground. And here, the surface level of the soil is usually just the organic matter. Thus, we think that it's important to sample the deeper soil to test the concentration of heavy metals in the soil. Furthermore, we should have also performed a soil test before our experiment. And as Maria mentioned, our main objective is to verify if the plants are good hyperaccumulators. In other words, if these plants absorb heavy metals from the soil. However, how can we really detect the metals from the soil if there were no metals to start with? So by performing a soil testing, we can initially detect the potential toxicities that are present in our samples. And all of this, by analyzing all of this, this can really help us improve our future research.
Now, let us look at suggestions for our future experiments. By artificially creating an environment which was rich in our elements zinc and cadmium that we were interested in, we could better observe the hyperaccumulation in our potential hyperaccumulator plants. Secondly, since our plants were collected in winter, that means many metabolical activities were dormant. But as Maria mentioned, these plants are perennial in nature and grow throughout the they grow throughout the year, indicating that we could perhaps try comparison during another season, such as spring, when more metabolical processes are going on, and allow for comparison. And also, yarrow plant is often consumed in teas, and as our data suggests that there is an accumulation of zinc in these plants. And since zinc is also essential not only for plants, but also humans, we could also explore the human health benefits of yarrow extract consumption. Lastly, an interesting observation was that we found many more toxic heavy metals accumulate in the gym soil as compared to the D soil. The gym soil is next to a huge parking lot, indicating that a possible reason for this could be the car emissions causing car pollution, which we could further explore.
So in conclusion, our data gave us the impression that these plants are selectively taking the elements that they need to start growing into microelements. They are not uptaking, they are selecting the elements and not uptaking from the soil. For example, the soil showed that it was copper on it, but the plants weren't absorbing the copper from it. Also, the same graphs they gave us the impression that these plants are giving us the same, they are taking the same molecules that they have in all plant organs. So it doesn't matter the location where they are, they're still behaving the same way. We cannot conclude that these plants are hyperaccumulators, but if you expose them, they might behave in this way. We have to do further research in order to answer a question, and we might reform the questions as another way. However, phytoremediation is still an environmental technique that we have to approach by using these plants, because due to their lowest cost, the benefits that they can give us, not only economically, environmentally, and also to our health. By having this, we know that phytoremediation is a good innovation approach that we might use that is at a lower cost, and we can benefit us in all Canada.
Well, after 7 months and finishing the project, I think this is the last time we'll be working on it. And I just wanted to thank everyone that helped us through the process. It's been 7 or 8 months. We used every advice you gave us because they were very vital to us working. We had our ups and downs in it, in our experiment and our presentation. And I really want to thank Ms. Lissy, Mr. Jailson, Amanda, Adam, David, Tracy, Mark, and our funders, NSCRC, for helping us go through this as a team, for working us and guiding us to reaching our potentials. Because I know we all know that from the beginning, we had a lot of ups and downs with our questions and how we reached here. I just want to thank everyone. I think there's
Any questions?
in the chat.
There's something in the chat.
Oh, thank you.
Well, thank you guys very much. That was an excellent presentation.
Thank you.
Thank you.
Amanda, do we have time for any questions? Does anybody on the call have questions? Okay, well, I have a question that I tend to ask all of the student groups. I don't know if you have time to answer it though. If you do not, I'll ask if I can send it through Amanda via email to an answer from it. You told us lots about your science experiment and the results that you learned and everything else, and you did a fantastic job. It was really, really good. I'm curious about you as a group of students that have gone through this experience and what you learned about yourselves that you would carry forward into your future school or work world.
One of I think the best things about this project was that we were supposed to do everything. We've been here in 2 years at Vanier. And for example, the closest thing we have are labs. But when we go to a lab, we have already the lab manual. We have everything we need. We have the procedure. We're just there to do it and find our results. But when we came here 7 months ago, we were all in a class with different ideas, different mentalities, different in every way. And we had to all come and agree on one question randomly. And there was nothing to help us. Well, of course, there was the teachers there to help us. But at the end, it was more of student-based experiment, which was, I think, the best thing that helped us think that, yes, with imagination, with our work, with our research, it made us push our limits into finding more about ourselves, into finding, researching, or maybe looking at what we would love to do or what we like to do in the future. It was something that was very helpful. And because it was just us working on it, we had to believe in ourselves, of course, with the help of all the teachers that were there. But at the end, it was a student job, a student project, and we came up with the question. We worked on the question. We had to find the procedures, and we just had to trust in each other as a group and work as a group. And I think that for me, I would say, helped me a lot because I was working with a group of people that all had the same interest. We were all interested in the same project, and that could help us in the future when you work with different people in the same jobs, different jobs. you're working with different people around the same thing with people are thinking this, like thinking very closely to what you want, and that motivates you to work harder. I think that was a very good experience.
I also have something else to add.
Yes,
Something else I
please.
know
I'd
about.
like all the students to come and talk to me if you could, if we have time.
Was that it's often worth still pushing on with the experiment. There were oftentimes things didn't go exactly as planned. We started off with a hyperaccumulation study and ended up proving, well, not proving, but showing data that indicated something else. So that didn't just happen in the general project, but also it happened, I know, to me, literature research. I'm looking for one piece of data and then I come upon something else that will also be helpful for another aspect of the project, or it could help us ask a new question or view it from a different angle. That's something I know from this project.
Thank you.
Something that I learned from the project was at early beginning, we didn't have phytoremediation at the beginning, to be honest. It wasn't mentioned in the presentation. At first, we were looking for some nets that basically they're neutrophil extracellular traps, which is the immune system. We have to grow amoebas and know how to, we needed like ethics and everything, documentation, and we were able to do this. So we had to come into this idea, and just by thinking about what was around us, we got into, okay, let's do something about the plants at the gardens. And with the help and the suggestions, we got into this. So we basically learned how to adapt into a time frame. We also had to adapt ourselves to the limitations we had because we didn't have any more time to still think about it. But as well, we also switched roles with the teachers. We were not only students anymore because we were conducting ourselves into this project and coming up with everything. We learned how to do properly literature review. We always have these reports of class. Yes, we have to do reference and everything, but going into a new topic that it wasn't imparted beginning from a teacher, you don't have any introduction to it, and going into these papers and like not knowing what words they were written on, that was a part like we would struggle most. We didn't know what the terms of the science they were using, and we were always wondering, what is this? We Googling every small word that it was because we didn't understand everything. And we got into the idea, okay, we can even understand the next articles. Why we are taking something that we are not able to do it? Not yet. Maybe in the future, but not yet. So we were just like going back to realize it, okay, this is a learning process. So we have to go into small steps so we can go someday into the summit, let's say. So we learn how to take a small, everything to take into account everything that was around us, and how to learn from research and also putting in practice what we already learned in college through the teachers. So it was basically blending everything, the new stuff, our strengths as well as the weakness, and we were supporting each other. We were sending information to each other, so we were able to just communicate a bit more so we can understand everything. And also by working and learning it in a whole group, it's difficult, but we learned that, how we can communicate. We thought at the beginning we didn't know how to communicate properly. We were using Teams, the notifications were coming, nobody was answering the messages. So we just say, okay, we should move to something we use. We use what? Instagram, social media. Let's go for it. So that was something that came up, and it's something that we learned because we were just focusing like we didn't know ourselves, we didn't know each other. So we learned that too, and now basically they got us into know most on other peers that we might also encounter in other classes too. Thank you.
Very good. Thank you.
What I would like to add was, what I had learned was like the very hardest part of the project was the beginning. Like as much as we starting with a question and then kind of get the results, we were a little disappointed, but just starting, honestly, that was like the hardest part of the project. We were like, okay, we all decided we would come up with 2 to 3 ideas. We meet up and we realized that some ideas are really different and some of them, we don't know what to do with it yet, and we didn't sure, is this something possible? Has someone done it already? Or is this something someone already talked about, but no one's proved it? So now it's picking a direction and, focusing on it. And the first 2 months, I gotta say, the first 2 months were just that. We're just picking a direction and then we were going with it. And it's like, oh, we hit a block. I'm like, okay, let's try another direction. And then, that direction didn't work. Let's go back to the original one. And we kind of jumped from idea to idea. And like, it was, I gotta say, a little frustrating, but it's satisfying to go like, oh, we have an option. We have like a path we could take. And that is something that I learned, is that even if I hit a few blocks, I just step back and look around at the other options that I took and maybe I didn't really want to do, or maybe that they didn't just pop in my head yet. So, and that's the thing with the group, is that something that didn't necessarily come from me, someone else mentioned it and they're like, wait a minute, that's a great idea. And we just bounce off ideas from one to another. And I really like that, at the beginning we were kind of a little shy getting to know each other. And after a couple of months, it was kind of like, hey, can you do this? Sure, let's do it. And I guess that's how you kind of work when you go in a career. You got to work with people, right? Even if you live alone, you have to present something to someone. And when you do that, you have to bounce off ideas that maybe didn't come from you. And sometimes that's the best ideas, right? The best ideas come from a group of minds that come together. And it's fun.
Great, thank you. Thank you.
So what I learned is the importance of collaborating with experienced people, and that it's always that you must always be teachable. Because at first we were doing analyzing a research about amoebas, and for us we're like beginners, so we're kind of thinking of hard experiments that might not be weren't even possible for us. So it's just important to work with experienced people, like for example, Amanda, Miss Lysian, Mr. Jailson, and also with Dr. David Moore. Like they, all of them, like they invested their time on us, like helping us, replying like every single time. And yeah, so they help us. So yeah, I think it's really important to have a mentor. Okay, thank you.
Great, thank you. Thank you very much.
So for me personally, just mainly that I learned how to be more patient and cooperate better, because we all have different schedules. So we don't all have the same amount of time we can work on it. We can't always work on it at the same time. So sometimes one person has their stuff done, but then they need someone else to check it over, but that person can't check it over until maybe 2 or 3 days later. So then during the meantime, they do more research and they change stuff again. You just need a lot of patience and a lot of communication, which we didn't initially have at the beginning. I mean, we didn't really have it until I think February. But yeah.
Thank you. That's a great observation.
And lastly, I would like to say, I think it taught me a lot about the research project, how it's not always like a linear path. You got curves, you got twists and turns, and so many dead ends. I think it taught me how to take in disappointment for once, because we were so passionate about the amoeba project, how there are these DNA nets that have links to our human immune system as well, and we could perhaps cure cancer.
Spectacular.
So it taught me, however, so it really did teach us how to take in disappointment, as well as how to work as a team. It taught us a lot about team dynamics, how there are different personalities and how communication is important. And honestly, it was an amazing opportunity.
Great, thank you. I'd like to invite the teachers to come and share as well, if they can.
Hi, Tracy. So we don't have a lot of time, but I'll quickly share, I guess, the most important point for me. I think it's really letting go of control over a group of students, because normally when you teach, you have everything, especially in science, we have a lot of like content heavy classes. We have everything planned to the minute almost. everything that is going on in a class and the assignments and everything. And in this project, it's very different because the students take control over the project, their timelines, the like the procedures they are going to do. So it's really student-led. And it was a very, very interesting experience for myself to kind of step back and let them make their decisions and get organized and just being there to support them. So it was very interesting. I think it was a great learning experience for me as well.
Thank you. Thank you, all of you. You did a fantastic job. Thank you for your time. And yeah, I hope you're all very proud of yourselves because you deserve it.
Yeah, so recognizing that we're short on time, students do have to catch to their next class. So yeah, just again, thank you for everyone and those of you in the room for attending. Really well done, students. I'm quite proud. You definitely came a long way. It was a really good presentation. And yeah, we'll touch a base again soon, following up with some other stuff. But just, yeah, definitely pat yourselves on the back. That was really well done. And thank you. I'll stop the live streaming and we can leave here.